All living organisms has a unique characteristic, which is to produce organism of its own kind. The production of new individuals from their parent is called reproduction. We know that a flowering plant consists of different parts. Different parts of the plants are classified into vegetative and reproductive parts, and each part is responsible for certain function. Vegetative parts are responsible for the development and nutrition of the plant. Vegetative parts include leaves, roots, leaf buds, and stems. Although the vegetative parts are not directly involved in seed production. Second category is the reproductive part. These are involved in the production of seed. They include flower buds, flowers, fruit, and seeds. There are different ways by which plants produce their offspring. These are categorized into two groups: asexual reproduction and sexual reproduction. In sexual reproduction, two parents are required, that is, male parent and a female parent. The two plants produce gametes that fuse together to form an individual. In sexual reproduction, new plants are obtained from seeds. It is the most common way of reproduction in plants. The second category is called the asexual reproduction. In this type of reproduction, the plants do not form any seeds. The vegetative parts of the plants, like stem, root, or leaf, give rise to the new offspring. The offspring looks exactly equal to the parents and have no genetic variability. The young ones which are formed are almost identical to each other as well as to the parents. There are different modes of asexual reproduction, for example, budding, spore formation, fragmentation, and vegetative propagation. First in the list is budding. We know yeast is a single-celled microscopic organism. These grow and multiply within hours if sufficient nutrients are made available to them. Let's find out how they reproduce. The parent yeast cell develops an outgrowth at its upper end. After a while, the nucleus of the parent cell divides into two, and one of them moves into the outgrowth, which is called bud. This bud grows bigger and bigger and finally separates from the parent cell into an independent new individual. The new yeast cell grows, matures, and produces more yeast cells. Sometimes another bud arises from the bud, forming a chain of buds. If this process continues, a large number of yeast cells are produced in a short time. This whole process is called budding. The second mode is called fragmentation. You might have seen the slimy green patches in ponds or in other stagnant water bodies. These are algae. When water and nutrients are available to these algae, they grow and multiply rapidly by the process called fragmentation. Fragmentation is normally seen in multicellular organisms which have relatively simple body organization. Spirogyra has a filamentous body. It breaks up into two or more fragments. These fragments or pieces grow into new individuals. Next is spore formation. This mode of reproduction is seen in RHIZOPUS, which is a fungus. RHIZOPUS body is made up of thread-like structure called hyphae. Each hyphae has sporangia, which contain the reproductive structures called spores. When spores are released, they keep floating in the air, as they are very light and can cover long distances. Spores are asexual reproductive bodies, and each spore is covered by a hard protective coat. To survive the unfavorable conditions like high temperature and low humidity. So, because of this feature, they can survive for a longer time. When these spores get favorable conditions, a spore germinates and develops into a new individual. Plants such as moss, ferns also reproduce by the means of spores. Next category is vegetative propagation. It is a type of asexual reproduction in which new plants are produced from roots, stems, leaves, and buds. Since reproduction is through the vegetative parts of the plant, that's why this method is called vegetative propagation. It is again divided into two types: natural vegetative propagation and artificial vegetative propagation. Natural vegetative propagation happens when plants grow and develop naturally without human intervention. For example, tuberous roots of sweet potato, asparagus become swollen due to the storage of food. This forms a new plants in the next season. Tuber of potato is an underground stem which stores food. It has buds in the depression called eye, which can give rise to a new plant. Rhizome is also an underground stem that has buds, for example, ginger and turmeric. Bulb is a small disc-like stem surrounded by scale leaves. It is found in onion. In this, a new plant can arise from the stem. Bryophyllum is also called sprout leaf plant. It has buds in the margins of the leaves. If a leaf of this plant fall on a moist surface, each bud can give rise to a new plant. Other than this, plants such as cacti produce new plants when their parts get detached from the main body. Each detached part can grow into a new plant. Second method is called artificial vegetative propagation method. These methods are frequently used to give rise to new and sometimes some novel plants. They include grafting, cutting, layering, and micropropagation. Some plants like money plant, rose are propagated through stem cuttings. In this method, the healthy branch having leaf buds is planted in the moist soil. The cutting develops roots and grow into a new plant. Second method is called layering. In this process, the lower branch of the plant is bent towards the ground and covered with moist soil. The tip of the branch comes out of the soil. After some days, the roots are developed. The branch is cut and allowed to grow in a new place. Young stems that can be bent easily without any injury are preferred. Jasmine and bougainvillea can be propagated by this method. Another method is called grafting. Grafting is a common method used to develop new varieties. In grafting, two plant species are used. Part of the stem of the desirable plant is grafted onto a rooted plant called the stalk. The part that is grafted is called the scion. Both are cut at an oblique angle and then are held together, matching up these two surfaces as closely as possible. The vascular system of the two plants grow and fuse together, forming a graft. After a period of time, the scion starts producing shoot and eventually starts bearing flower and fruit. Another method is called the micropropagation or plant tissue culture. It is a method of propagating a large number of plants from a single plant in a very short time under laboratory conditions. This method allows propagation of rare and endangered species that may be difficult to grow under natural conditions. To start plant tissue culture, a part of the plant such as leaf, stem, embryo, anther or seed can be used. The plant material is thoroughly sterilized. Under sterile conditions, the plant material is placed on a plant tissue culture medium that contain all the minerals, vitamins and hormones required by the plant. The plant part give rise to an undifferentiated mass known as callus, from which individual plantlets begin to grow after a period of time. These can be separated and are first grown under greenhouse conditions before they are moved to field conditions. Vegetative propagation allows quick and easy propagation of the plants. Plants can bear flowers, fruits earlier than those produced from the seed. And it is only possible method of reproduction for such plants which have lost the capacity to produce viable seeds. In case of vegetative propagation, the offspring is an exact copy of the parent. Hope you enjoyed today's lesson. Please like, share and subscribe to my channel. Thank you for watching.